Good afternoon, everybody. And it's a real pleasure to be here and to have the chance to tell you about our efforts to attack MYC using cell-penetrating mini-proteins. You will hear about MYC several times during this conference, so I just want to convey a message that MYC is an unusual oncogene because compared to other oncogenes that we often hear about, like KRAS or PI3 kinase or HER2, for example, MYC is not often found mutated in cancer. Most of the times, it's actually deregulated by upstream oncogenic signals. So these lesions I just mentioned, KRAS, HER2, or PI3 kinase, all funnel through MYC inside the nuclei. MYC is essentially their culprit, their minion, that execute all the transcriptional programs that then lead to tumorigenesis. MYC is a master regulator of all the transcriptional programs that can lead to cell proliferation, to the coordination of the crosstalk with the tumor microenvironment. And it's been recently shown that MYC is also responsible for immune tolerance. So it's what makes cancer cells invisible to our immune system. Despite all these roles of MYC in cancer maintenance, there is still no MYC inhibitor in the clinic yet. And the reason is that for a very long time, MYC has been considered an undruggable target. And the reasons have been various. We know, for example, that MYC is an intrinsically disordered protein. That means that it has a structure that changes all the time in solution, so that the standard small molecule approach has had some difficulties to inhibit it with sufficient specificity, but you will hear more about that during this conference. The other reason is that it resides in the nuclei of the cells, a compartment where a lot of drugs do not get. The other reason is that it doesn't have an active site, so it's not like an enzyme with an enzymatic pocket that can be inhibited easily. And the MYC family comprises three different proteins, c-MYC and MYC and del-MYC, that have been shown to be able to replace each other in some situations, so that if we want to inhibit MYC, we better inhibit them all. And last but not least, it's been thought for a very long time that since MYC also has a role in normal tissue maintenance and regeneration, inhibiting it would cause catastrophic side effects in normal tissues. However, when I started working on this more than 20 years ago as a student, I basically found no evidence of the fact that somebody had even tried because of fear of these catastrophic side effects. So I decided to do it at least in vitro. And in order to design how it was possible to inhibit MYC, I focused on this specific biological aspect of MYC. In order to function, MYC has to dimerize with its partner called MAX. And when it does, its C-terminus assumes this wonderful three-dimensional structure here shown on the right, which is the BHLH zip domain. So these nice forceps are able to bind DNA, and this is how MYC exerts its function as a transcription factor. So I decided that I had two options: either inhibit MYC-MAX interaction or inhibit their interaction with the DNA. And in order to achieve that, I designed this first dominant negative of MYC that I called HOMO-MYC. HOMO-MYC is essentially a small portion of the entire MYC protein. It's the BHLH zip domain only, but it has 4 amino acidic substitution in the leucine zipper. That means that while MYC can only form heterodimers with MAX, HOMO-MYC can do much more. HOMO-MYC can form heterodimers with MYC, and these heterodimers are not able to bind the DNA anymore, but it can also form homodimers and heterodimers with MAX. These two dimeric forms are able to bind DNA, but they are silent dimers. They don't have any transactivation domain. So they basically shut down the transcription of these target genes. So HOMO-MYC, in a nutshell, has a different mechanism of action to function as a dominant negative of MYC. It has the ability to do active sequestration of the target, but it can also compete for the binding to its target genes. Amomic has been validated over the years as a pan-MYC inhibitor. It's been shown to be able to inhibit c-MYC and MYC and L-MYC, so the old family. The big shift in paradigm regarding MYC druggability was achieved when we managed to clone Omomic in the first systemic mouse model. So in this case, we put Omomic under the control of a tetracycline responsive element. So these mice were then crossed with the reverse tetracycline independent transactivator under an ubiquitous promoter, which meant that Omomic was normally not expressed in the mouse until we added doxycycline to the drinking water of the animals. At that point, Omomic was expressed systemically in all the tissues of the animal. And this was our way to model systemic inhibition of MYC, to model a first drug against MYC. And in order to show its therapeutic effect, we crossed these animals with a very well-known lung tumor mouse model. This model characterized by Dave Tueson in Tyler Jack's lab. And basically, it's a model in which endogenous KRAS has been replaced by a mutated KRAS G12D, preceded by a lock-stop-LOX cassette. This cassette can be removed by Cre recombinase. So if we provide the animals with adeno-Cre intranasally, KRAS gets selectively activated in the respiratory tissue of the animals that develop hyperplasia and adenomas by 4-6 weeks after the intranasal instillation, or carcinomas by 16-26 weeks. And here we tested the therapeutic impact of Omomic. We basically let the tumors develop. These animals develop hundreds of tumors in their lung, and we turned on Omomic with doxycycline added to the water of the animals. And only 3 days later, we observed this dramatic therapeutic impact. All the lesions had shrunk. So this was mind-blowing to us. We expected maybe a cytostatic effect. We really didn't expect this shrinkage of the tumors. And even more impressive, 1 week later, the tumors were all gone. This, of course, conferred a very significant survival advantage to the animals that were treated only once with Omomic, as you can see here. But more impressively, animals treated metronomically with Omomic stayed tumor-free indefinitely. So we seem to have found a function in lung tumors, MYC, that seemed to be absolutely non-redundant and around which tumors could not evolve. But the biggest surprise, possibly, was not just the therapeutic impact of MYC inhibition, but the fact that it presented an unexpected and excellent therapeutic window. Everybody expected the normal proliferative tissues to suffer from MYC inhibition. And instead, what we saw was that all secondary effects were absolutely mild and completely reversible. The proliferative tissues, normal proliferative tissues, simply slow down their processes. So we would observe slower wound healing, for example, slower hair growth in case we shave the animals, temporary, only transient anemia compensated by exolocation of hematopoiesis to the spleen, less fertility during the treatment that went back to completely normal once we stopped the treatment. And maybe the most impressive phenotype was the intestine, where we observed that the villi became a little bit shorter during the treatment, but the intestinal lumen compensated, increasing and creating the same absorbing surface, so that at the end of the day, all the blood chemistry showed completely normal values. So this was really a shift in a paradigm. We published this in Nature in 2008, and it was the first proof of concept that MYC inhibition was feasible, had an impressive therapeutic impact in tumors, and it was very safe in normal tissues. Since then, Omomic has been transgenically used in many more mouse models of cancer, in which it showed very dramatic therapeutic impact independently on the tissue of origin of these tumors, or the mutational profile of these tumors, again reinforcing the notion that MYC has functions as an essential node downstream of all the oncogenic lesions that we can think of. But again, this was done for decades using MYC as a transgene. Can MYC itself be a drug? Here we encountered possibly the biggest challenge because it was a renowned notion out there that MYC was just a proof of concept. There were reviews stating that it was a molecule too big and bulky to be directly delivered to cells. So it was bound to be a proof of concept and it could only work as gene therapy. However, in 2012, Pierre Lavigne in Sherbrooke published this interesting paper showing that the max BHLH CYP domain, which he called MAX-star, was able to transduce cells spontaneously and exert some anti-MYC transcriptional activity. So we thought that MAX-star and MYC were very similar, not only in terms of sequence but also structure. And so we thought that if MAX could penetrate cells, maybe MYC could do it too. If not, we could modify MYC to make it more similar to MAX and achieve the same result. So I had the enormous luck to be able to hire somebody from Pierre Lavigne's lab, Marie-Ève Beaulieu, who came as a postdoc to my lab, and Marie-Ève started producing MYC as a recombinant protein instead of as a transgene. And to our really pleasant surprise, we saw that when we added MYC, in this case labeled fluorescently with PITC, MYC was able to penetrate quickly the cells and reach their nuclei, exerting its anti-MYC activity. It would do so through different mechanisms of entrance. We used several endocytosis inhibitors, and we showed that MYC can penetrate cells through mainly clathrin-dependent endocytosis and macropinocytosis, but there are other mechanisms as well. And this is exciting to us because the more mechanism of entrance we have, more cells we can penetrate independently of different receptors that these cells might have. Not only macropinocytosis is enhanced in cancer cells, which means that MYC will have an advantage to penetrate cancer cells compared to normal cells, hijacking this system that is already increased in cancer cells. And once it's inside, MYC basically arrests or causes death of these cancer cells with an IC50 in the low micromolar range. Here we included as a control SHEP cells, which have been described in the literature to be less dependent on MYC. And indeed, we need to increase significantly the amount of MYC in order to see some effect in these cells. But again, we had to show that this was an on-target effect, that we were pumping cells with a protein potentially big and bulky, right? So we had to show that what we were seeing was MYC dependent. We looked at MYC by ChIP-seq analysis. These are bona fide MYC target genes I'm showing you here, and in black, you can see the nice binding of MYC to these target promoters. And in green, you can see the nice effect that MYC has on this binding. In this case, these were A549. We saw this displacement from DNA without any change in total MYC level. And this can be done also at genome-wide. And so you can see in red here, the binding of MYC around the transcription starting site of several of its targets. And this is the effect seen by MYC. We calculated the extraordinary effect of MYC to be displacement of MYC from more than 97% of its target genes. And this, of course, translates in a very impressive transcriptional reprogramming of the cells. These are 3 non-small cell lung cancer cell lines, but we have seen this in other tumor types as well. You can see the shutdown of the classical MYC fingerprint. So this is really an on-target effect, corroborated also by the fact that instead other BHLH ZIP transcription factors are not affected at all. These transcription factors have been reported to be involved in lung tumorigenesis, but in this case are not affected by the treatment with MYC. So encouraged by these in vitro results, we decided to go in vivo and test the therapeutic impact of Omomic there. To start with, we decided to try local administration. We wanted to target lung tumors, so we went for intranasal administration of Omomic. In this case, we used a fluorescent Omomic to be able to track it. And we saw that after 1 hour, all the lung was positive for Omomic, but also other organs were positive, such as brain, liver, pancreas, kidney, intestine. And this suggested that through the bloodstream, Omomic was able to travel to these other organs. This was already very, very interesting to us. But in order to focus on its activity on lung tumors, we treated mice that carried lung adenocarcinomas. And again, to our surprise, we saw that 24 hours after the intranasal administration of a radioactive Omomic, Omomic was accumulated specifically in the lung tumors, while it had washed off from the normal tissue in the lung. So it looked like it knew where it was supposed to go and accumulate. Here it's the dose at half an hour observed in the lung, and this is 48 hours after the intranasal administration. You can see that the lung is still very positive for it. So what's the biological effect of this administration, intranasal instillation? So we followed animals that had adenocarcinomas in their lung for 4 weeks, either treated with vehicle or with Omomic. The vehicle-treated animals during these 4 weeks showed all tumors that increased in size or even appeared de novo. In the Omomic-treated cohort, instead, we didn't observe any significant growth of the tumors. We thought that this was an amazing cytostatic effect, but the pathologist made us notice something more interesting than that. As I said, the vehicle-treated animals, as expected, have the majority of adenocarcinomas. But the Omomic-treated animals, instead, in more than 50% of the cases, have regressed to being adenomas or simply hyperplasia. And this is the result of both reduction in tumor proliferation, but also increased cell death as seen as cleaved caspase 3. We also observed something really interesting, that these tumors that are KRAS-driven tumors, notoriously immune deserts, become significantly infiltrated with T cells upon the treatment. So for the sake of time, I will not show much more regarding this aspect, but you can imagine that this has prompted our group to test the combination of Omomic with immune therapy. But in order to unleash the complete potential of Omomic, we wanted an intravenous product that could reach not only the primary tumors, but also its metastasis. And in this case, we started with one of the non-small cell lung cancer tumor models that we had in the lab that was the most difficult to treat. We had the H1975 human cell line that is EGFR mutant, PI3 kinase mutant, P53 mutant, and it's resistant to the standard of care for EGFR mutation. Lotinib in this case. So we decided to try Omomic in this tumor that doesn't respond to basically anything that we have in the lab. And we saw this very nice response. This is something that we observe often. The tumors initially slow down, and by the 3rd and 4th week of treatment, they start crashing. We were very, very pleased to see that Omomic displays a half-life of 49 hours after IV administration. This was, again, quite surprising because peptides or mini-proteins have been described to have a half-life of usually 20 minutes only. In this case, we used once-a-week administration of 30 milligrams per kilo of mini-protein. I want to also underline that this therapeutic effect is not limited to non-small cell lung cancer only. Now we have a nice proof of concept also in triple-negative breast cancer and other types of tumors. In this case, we injected triple-negative breast cancer cells, human cells, in bulk sinew mice, waited for the tumors to appear, and then we started treating with Omomic. And we saw this very nice therapeutic effect. One thing that got us really excited is the possibility of intervention and prevention of metastasis. A 3-day pre-treatment of these same cells inoculated by tail vein. These cells usually colonize very efficiently the lung and for metastasis there. These 3 days are sufficient to prevent these lung tumors from appearing and growing. So we are really, really hopeful that this could be another interesting application for Omomic. I don't have to tell you that our overarching goal is to provide a viable therapeutic approach for intervention in cancer, and we hope to offer a more efficient and less toxic therapy than many therapies that are out there. And this is why, with Marie-F, we decided to fund Peptomic in December 2014. And we are now completing our preclinical safety studies in rats and monkeys, and if everything goes well, we'll be able to start clinical trials, first-in-man studies, in Q1 2021. With MIC, we could go in multiple types of tumors. We have selected so far triple-negative breast cancer, KRAS mutant lung cancer, and RAS mutant colorectal cancer, but we are evaluating other types of indications as well. With this, I will end acknowledging all the people, both in my lab and Peptomic, they really work side by side that contributed to this, and our amazing collaborators around the world. I will be happy to take your questions, but before then, I would like to show you really the faces of the people that did the work. Thank you very much.